Thank you very much. So my collaborators and I would like to report the surprising finding. The top-down feedback from the ventral stream of the visual pathway to V1 is focused on V1's representation of the central visual field. And this was predicted by a theoretical framework. So along the visual pathway, the visual inputs are feed-forward and then feedback. The ventral stream is more specialized for visual recognition, whereas the dorsal stream is more specialized for locating the objects. So if we zoom to V1, one patch of V1 surface is very similar to another patch. And traditionally, the V1 surface is seen as uniform in their neural properties, such as the sizes of the hypercolumns, they are uniform across the surface, and the cell densities and the sizes of dendrites and axons and so on, and the thickness of the ocular dominance columns, and the neural tuning properties to visual features like orientation, for example. Meanwhile, this patch is for a more central part of the visual field, so its neurons have smaller receptor fields, and this patch is for a more peripheral visual field location, so the receptor fields are larger. Nevertheless, as long as the visual inputs are scaled to the sizes of these receptor fields, V1 surface has been seen as uniform in its neural properties. Now, more specifically, for a retinal input, it is mapped to the V1 surface, and a foveal patch or a peripheral patch receives input from a smaller or larger region of the visual field. So the central visual field is magnified more. Again, other than such a differential magnification factor by the feed-forward transformation from the retina to V1, the V1 surface has been seen as uniform. For example, per square millimeter of V1 surface, the number of feed-forward inputs from LGN is roughly constant. However, we found that the feedback from the ventral stream is denser to the central visual field, so that a square millimeter patch nearer to the fovea receives more feedback projections. This can be quantified by the feedback magnification factor, which is the number of feedback neurons per feed-forward neuron from LGN. So now let's focus on this feedback and to measure this magnification. To do that, we injected retrograde tracers to various V1 locations. The tracer works like this for a neuron. The injected tracer is absorbed by the axon terminals and is transported to the cell body so that one can label and identify by the color of the tracer this presynaptic neuron. And this way, the feed-forward neuron, the feedback neuron from outside V1 can be identified, and you can even count how many of them, for instance, in V2. And similarly, you can label the feed-forward neurons from LGN, and therefore also count their numbers. And the feedback magnification, for example, if you focus on V2, is then the ratio between these two numbers. So let's say if you, this is an example. This is an example of the cortical brain unfolded, flattened for a marmoset monkey. And this is an injection site in V1. And these black dots visualizes the neurons in V2 that feedback to this injection site. And you can count the number of these neurons when you slice the brain into thin slices, such that individual neurons can be individually identified, and you can count them clearly. And but if you inject to a different location, let's say it's a V1 patch closer to the fovea, and then the prediction is that this feedback magnification will be higher. And so let's look at the results from details. And this is the feedback from V2. We have 12 injection sites for visual eccentricities from 2 degrees to 18 degrees. And the magnification, feedback magnification dropped by a factor of 10. And you can also look through V4, IT, and all these ventral stream cortical areas, and we just pull all these feedback neurons together. And again, the same trend is seen. A factor of 10 drop from 2 degrees to 18 degrees of eccentricity. But you can also do this for the dorsal streams, and you find that this trend is less steep. And we will explain later why. And excluding V2, now here, the ventral and dorsal streams are all excluding V2. And so we'll continue to look at excluding V2. You can see that for the central visual field locations, the feedback is dominated by the ventral stream. However, for the peripheral locations, it's the dorsal stream that dominates the feedback. And so therefore, V1 is far from being a uniform surface. There's qualitative changes between central and peripheral. And so therefore, when you look at a neural circuit, it's not only as a function of the hierarchical level along the visual pathway, we should also look at another dimension, which is central peripheral dimension, and the visual circuits for the central visual field and for a peripheral visual field may be qualitatively different. And this is actually predicted from this central peripheral dichotomy theory in the looking and seeing framework. Traditionally, vision is viewed as just seeing what is where in the visual field. And however, it is an illusion that we see the whole visual field clearly. An attentional bottleneck makes us see clearly only the part near in our attentional spotlight, which is usually centered at the fovea. So the question is, where shall we shift our gaze next? Next, where do we go? And this, to address that, we have this new framework in which vision is both looking and seeing. And looking really is to try to select a fraction of visual input into the attentional bottleneck, mainly by the gaze shift. Specifically, given the visual image, it is actually V1 guides looking exogenously by creating a bottom-up saliency map of the visual field. The salient locations in this map attract gaze shifts reflexively, guiding attention exogenously by the external input. And you've heard this also in Wuli's talk. So then the bottleneck starts from the output of V1 to downstream areas where seeing happens. What is seen is centered at the fovea by the gaze shift. So therefore, the central visual field is specialized for seeing, whereas the peripheral visual field is specialized for looking, to decide where in the visual field to direct the gaze. And even in the central visual field, the bottleneck can limit the initial feed-forward information so that visual perception can still be ambiguous. In such a case, a feedback to upstream areas, such as V1, can query for additional information needed to disambiguate and verify perceptual hypothesis. So for this example, if the initial feed-forward information is insufficient about the shape of this object to distinguish between a flower and an apple, then the feedback can query for more shape-relevant information to disambiguate. So therefore, this feedback is also mainly directed to the central visual field. So these are the three components proposed by the central peripheral dichotomy theory in the looking and seeing framework. And so therefore, this theory predicted our new anatomical finding, since the ventral stream is specialized for visual recognition. On the other hand, since the dorsal stream is less specialized for visual recognition, its feedback to V1 is less sensitive to the visual field eccentricity, as observed in our data. The peripheral visual field lacks the feedback query, and so therefore makes seeing more difficult. For example, it gives visual crowding in the peripheral visual field, where cluttered inputs are more difficult to recognize. This difficulty in seeing also makes more than just visual crowding. It also makes errors in perception, which is visual illusion. So in this example, for example, you will see illusory flashes in these white disks, but these illusions are only visible in the peripheral visual field. So wherever you fixate, it's not flashing. Yeah. And you can also see this in this example. This is a static image, or these illusory motions are actually not where you are fixating at. Wherever you fixate, it's not moving. And so in a sense, that means the illusion is actually vetoed in your central visual field because you have additional queried information. And in addition, the feedback query also makes the white kinetic triangle here appear more vivid, because this feedback query does the computation of analysis by synthesis. So if you view this kinetic triangle peripherally, it's less vivid or less visible. Okay, and it also helps you to see a duck in this image, if you have a hint. So the feedback is very useful in the central visual field, though not so much in the peripheral visual because it's not available. And the theory also predicts two other illusions. One is called reverse depth illusion, and the other is called flip tilt illusion. And they are only visible in the peripheral visual field, and they are predicted from our classical knowledge of how V1 neurons are tuned to orientation or to depth. But they are also, you need additional elements, which is the assumption in our theory that the bottleneck starts from V1, as well as the feedback query is lacking in the peripheral visual field. And further, it predicts that both illusions become visible in the central visual field by impairing the feedback query, and these have been confirmed experimentally. By backward masking the visual input, you impair the feedback. And these findings suggest that, so the illusion is vetoed not because the central visual field has higher resolutions by feed-forward expansion in the retinotopic mapping to V1, but it's because of the feedback, denser feedback to the central visual field to query for information to veto the initially misleading information. So therefore, we start with a theoretical framework, and that enabled us to predict non-trivial predictions about the neural circuit, that raising a challenge to the traditional framework of the fundamental things in V1 even. So a lot of fundamental ideas how vision works, and it's directly connected with perception, with lots of perception phenomena, and of course, action to shift our gaze. Thank you very much.